So Sean has had introductions already from Rich, so I don't need to continue with that. Just one thing to note, actually, Sean and I do go back somewhere. He was on the SAB for Structural Genomics, which is a company I started back in the day. So it's nice to catch up with him again after all this time. And he's going to tell you a bit more about the T4R2 story, and I'm looking forward to hearing it. So, Sean, over to you.
So I'm going to pick up where Rich left off. Everything he told you was done with sort of beautiful genetic logic. The mutants, the phenotypes, the linearity of the genetic code, what have you. And what they didn't need to know was what R2 actually did. And this is actually a fairly common situation in the history of molecular genetics, where we have a mutation that has a phenotype. It could be a genetic disease in us clinically. It could be a fly that suddenly grows an eye out of its leg or something. And then you are a million miles, you have beautiful genetics, but you are a million miles from understanding the molecular basis for what's going on to cause that phenotype. So R2 is a curiosity. Various people over the years have written about how it's a little bit embarrassing that we have all this beautiful genetics and we don't yet, even today, 70 years later, know the function of these genes. What I'm going to tell you is sort of use that. This is sort of a touchstone for me, for reasons that I'll explain, the R2 genes. But this is also a general story of how do we go from genetics to getting to molecular mechanism, and how we've gotten better and better at that over the years, especially with a set of techniques that I'm going to describe. You need to know a little bit about the biochemistry as we understand it from 70 years of work of the R2 and REX system. This is also part of why it's a touchstone for me. I was trained by Larry Gold and by this guy, David Parma, back in the 1980s and 1990s. And David Parma made it his life's work, especially in Larry's lab, to figure out the function of R2 and REX. So Rich told you that lambda, phage, there's another virus that integrates into the genome of E. coli, lives inside of E. coli, and then makes these two proteins, REX-A and REX-B, that somehow are a defense system against T4. And the way I want you to think about them for the purposes of today's talk is that REX-B is the booby trap. REX-B is a bomb. It makes a little channel that, when activated, punches a hole in the membrane and destroys a proton gradient that E. coli needs for life, so it's a suicide device. If T4 comes in, the bomb goes off, the cell dies, T4 can't get out. REX-A is somehow the trigger that lambda uses to detect the incoming T4. These are all shown as molecular biologist blobs. We don't understand how this happens, but somehow REX-A detects the incoming T4, activates the REX-B channel, kills the cell, and somehow that we do not understand, R2A and R2B prevent the bomb from going off. David Parma worked all this out in a paper in 1992, and the field has made very little progress on this since. This is what I look at all day. I'm a computational molecular biologist. I analyze DNA and protein sequences. I write software and algorithms for interpreting this stuff. As you know, we've gotten very good at sequencing DNA. We can sequence it wholesale. I was involved in the Human Genome Project. We can now do a human for $100, and it would take maybe an afternoon. This is the sequence of the R2A and R2B locus from phage T4. And as I bring that up, there's the ATG, there's the stop codon TAA of the R2A gene, there's the ATG and the stop codon of R2B, and some flanking genes from the phage T4 genome. You can see the sequence, and we know from the kind of thing that all the early molecular biologists worked out that now I can take that in triplets. So here is the R2B gene that most of the genetics was done with, and I can write that down, 939 base pairs of sequence from start to finish, and I can translate it in the triplet code that was worked out in the 50s and 60s, and now I have the amino acids. That string of amino acids, when made by the ribosome, that linear string is going to fold up into some protein, and that protein is going to do something. What I do, people like me do, is now turn that triplet code and the 3-letter code for the 20 amino acids into a very simple string of 1-letter codes for the 20 amino acids. Methionine, tyrosine, asparagine, isoleucine. I can just read that across. That, what we're looking at now, is almost the entire region that Crick and Brenner et al. Did their experiments in. This is 50 amino acids of the so-called dispensable region, where this sequence actually does not matter. This is actually weird. There aren't sequences in little compact phage genomes that don't matter to the phage. This was almost unique. They took advantage of this. You could frame shift this sequence, read it in the other frames, and for whatever reason, R2B would still work. So something is special about this 50 amino acids. I'm also using it as an example of 50, here's 50 amino acids. What do they do? The reason that I can do this is because genetics is based on digital information. DNA is digital information. I can read it like a string. I can try to interpret it like a string of letters. That idea weirdly goes way back. Ironically, the first place that people have been able to find it is in a letter by this guy, Friedrich Miescher. He's the discoverer of DNA. But we did not know that DNA was the genetic material until 80 years later. He thought, and other people thought, that the genetic material was proteins. So he was actually speculating about heredity via proteins. But whether it's DNA or proteins, it's still letters in an alphabet, whether it's 4 or 20. And he said maybe that, in this letter, he said, maybe all this complexity is coming from some sort of language-like thing, arranging different letters like letters in an alphabet. And indeed, that turns out to be true. If I gave you a sentence of a language that you don't understand, like if you don't speak Swedish, but you saw that sentence, the way that you would try to interpret that sentence, not knowing Swedish, is by what we call comparative sequence analysis. You would say, well, I do know English, and I can sort of recognize some highly conserved words in there that are not changing very much from language to language. If you were a statistician and a computational person, you might take this to the extreme and start collecting all 6,000 or 8,000 of the world's languages and systematically compare how fast are the different words evolving over time, and imagine that you can transfer knowledge of a language that you do know to languages that you don't know with certain danger zones of like where there's close cognates that actually mean something completely different that you're going to have to experimentally test to make sure you're not making computational mistakes. But for the most part, you can see that as I go from languages that are closely related, more further related, and then far out, there's an evolutionary change with different rates for different words that you can see by lining up sentences that you observe in the languages. So we do this more or less with genetic material, and we have the difference with genetic material that we don't know the language. It's not like there's an English speaker telling us the meaning and we're just sort of mapping a new organism, a fruit fly, onto something else where we understand the language. But even in language, there are examples where people have collected a body of language that's been completely lost to history. And so a famous example is the language rIIB, which was recovered on stone tablets from Mycenae. Nobody speaks this language. It was unclear. It's an unknown script. But John Chadwick and Michael Ventris in the 1950s, working contemporaneously with Crick and Watson on a completely different problem, showed by statistics and computational analysis that if you have enough examples of your stone tablets, the statistics of comparative analysis of sequences is basically like cryptography. You can get information about the function or what the underlying language is. So we do that kind of comparison for biological sequences, for DNA and protein, and this again is the 50 amino acids of the dispensable region of R2B, looking at more and more distantly related bacterial viruses. The top sequence is the T4 sequence, and this is as we go to more and more distantly related things. I look at this, and it's sort of like looking at English, Swedish, blah, 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 And I say, whatever's going on here, this sequence is basically, it can't be changed by evolution. That is, evolution is doing a high throughput experiment for me. It has evolved a gazillion different R2 variants and tested them for whether they still work. And what I see as I sequence everything that's out there is I get the results of that experiment. Here's the things that work. I can infer that other things didn't work. And so I can look at this conservation pattern and I can say, that's really important, that EQ is really important, that's really important, this region doesn't matter much, this region doesn't matter much. Now, it happens that in this particular story, this is already a puzzle. This sequence is highly conserved across different viruses, but I already told you, and Rich told you, this is the dispensable region. It had to be dispensable for the Crick and Brenner experiment to work. T4 doesn't need this. You can mutate any of these residues, and T4 is fine with respect to the experiment that they were doing. So something is going on in the wild that T4 absolutely does need this sequence at the N-terminus of R2b for reasons to be determined. An advantage that we have over the stone tablets from Mycenae thing is that I don't have just 300 stone tablets from a cave. I've got the entire diversity of life. And we've gotten, like I said, very good at sequencing to the point that we have now sequenced about 1 million species. This is one of the more recent trees of all the species that have been sequenced. Most of them are bacteria. Most of life is bacterial. There are things called archaea, which are also unicellular, but are more closely related to us, to eukaryotes, than to the bacteria. Jack Shostak is going to talk about stuff that happened even before the last common ancestor. So back there somewhere, where we don't know where the root exactly is, it's the last common ancestor of all life. And then I want to impress upon you how broad this diversity is. That's you, that's mushrooms, that's corn. Most of what we think about as life, the diversity of life, if I were to show you a bunch of beautiful pictures of the diversity of life, is all sitting on this tiny little branch over here. And this is the rest of microbial life. All of that gives us information about the conservation of sequences over time. And I also want to impress upon you, we only think that there's on the order of maybe 10 to the 7th or 10 to the 8th species on the planet. We're still on an exponential ramp for sequencing. We are going to have the sequence of basically everything. Fortunately, though we have billions of protein sequences in the database, they are interpretable at more or less the same kind of granularity as words in human languages. Proteins are composed of domains. The domains shuffle between different sequences, especially in eukaryotes. And in sort of 100 or so amino acid blocks, different domains will have particular functions. And there's only about 30,000 protein domains, despite the billions and billions of sequences. 30,000 protein domains account for the vast majority of protein sequences. So if I get very good at recognizing protein domains, I get the equivalent of partial knowledge about words that are occurring in sentences of protein sequence. And so, for example, a molecular biologist can walk through this protein and say, oh, it's got a kind of domain that's involved in cleaving double-stranded RNA. It's got a kind of domain that's involved in binding double-stranded RNA. It's got a kind of domain that's involved in unwinding double-stranded RNA. And it's got some stuff that looks suspiciously able to do something with single-stranded RNA. And although you don't know the language, and so you're sort of struggling with your Italian guidebook to get by, you say, whatever's going on here has something to do with it's binding a double-stranded RNA, cutting it, unwinding it, and doing something with a single strand. And that is exactly what this particular protein does, which is a protein called dicer that's involved in something called RNA interference that won the Nobel Prize. So what I do day to day is I develop software and algorithms to let people in the community do this at scale. There's a software package called Hammer. The HMM part stands for Hidden Markov Model. And this was circa 1980s state-of-the-art for speech recognition. The parallels to language are sort of shot through this field. So using circa 1980s machine learning, speech recognition, mathematics, we now have software packages, including mine, in the community that can walk through a new protein sequence and assign it domains and therefore get partial information about its function. This has been industrialized. There's big databases of these. that systematically collate curated 30,000 protein domain families based on my software, and there's teams of folks at places like the European Bioinformatics Institute and the National Center for Biotechnology Information here in Washington, D.C. that do these things. And so, does that stuff actually work? So back to R2 as the example. Using 30 or 40 years of advances in our ability to do remote homolog detection, we've gotten better at getting little clues about systems like R2, these little mysteries. And so just to dive back into R2, we can tell by sequence analysis that these proteins do have domains in them, but many of them in gray are what we call affectionately domains of unknown function, DUFs, which is a reference to the Princess Bride, which has rodents of unusual size. And there's many DUFs that have been collated, but 3 of these domains actually do give me a little bit of information. There's a very clear ATPase motor sort of domain in R2A that I'd like to be able to sort of fit with some sort of mechanism. Weirdly, so this dispensable region keeps coming back. That highly conserved N-terminus of R2b is the only region of R2b that I can tell you what it does. It is absolutely a helix-turn-helix DNA binding domain. It's highly conserved. Why that would be dispensable, I have no idea. The rest of the protein, which I can't identify, is the bit that actually matters to Crick and Brenner. REX-A I don't know much about, but here's one that I do know something about, and it tells a little story about interpreting these kind of experiments for the computational experiments for biology. REX-B is homologous to a little proton-driven motor that's used to drive the E. coli flagella. So you may know that E. coli can swim. It does it by having a motor that can spin a tail, a flagella, and that's a really complicated molecular machine. As you look at all the different protein structures, I want you to concentrate on this thing, which is MOTE-A, which is a little spinning proton-driven motor. It lets protons through to generate its energy. It's anchored in the E. coli upper membrane with another protein called MOTE-B that gives it traction. Then when it spins, it engages with this rotor on the main flagella to spin it. Why is REX-B a bomb, but this is an intricate little piece of a machine? And one thing that evolution does is it says, there is a nice convenient hole that lets protons through there. I will throw away the rest of this beautiful machine and turn that little energetic thing that's capable of passing protons into a thing that I can use to destroy the proton gradient and become a bomb. So you borrow things that have a relationship and then deploy them, evolution deploys them in new contexts. So the word doesn't mean the same thing, but it has some relationship that we can learn from, much like we do with language. That's sequence analysis, and for a long time we would sort of get stuck with the kind of picture that I just showed you, where we would look and we would go, well, that's interesting. I've got a little bit of information, and it's a little evocative, but now where do I go? I am still a long way from biochemical mechanism. I am a long way from structure. And a huge revolution has now happened, which is the advent of AlphaFold. We're now at AlphaFold 3. Machine learning techniques using deep learning techniques. So everybody's heard about AI. This is one of the ones that's making a huge impact in molecular biology. I want to point out, because I'm very proud, that the input hiding over here in the figure to AlphaFold is my software. So the power of these multiple sequence alignments is that they have a ton of information about the constraints that are operating on those sequences. What AlphaFold's neural nets can do is take the output of my software or competing software from my hated competitors in Munich, who are actually quite good. Different pipelines are using one of the 2 software packages. Take that information from these multiple sequence alignments and see very subtle patterns of how the residues are co-evolving over time in alignments that are going typically tens of thousands of sequences deep coming out of these software packages and resulting in a protein structure. Remarkably, this is actually working. So the protein folding problem for single proteins and for small protein complexes is actually largely solved. We don't completely trust these methods, but they're amazing at the same time. And so nowadays, trying to hit the button to go back, I can just give a sequence to a server, the AlphaFold server. It runs my software, collects a multiple sequence alignment, immediately runs its neural net, and then I can say, look at gorgeous R2A, now as a protein structure, and that actually is structurally an ATPase, so it confirms what I thought from sequence analysis. It confirms the little helix-turn-helix DNA binding domain, the dispensable region. Here's the rest of the dispensable region, a little sort of fishhook line and hook here that's hanging off of the rest of R2b. God knows why, I'd still like to know. rIIB, the bomb, conveniently looking like a stick of 4 sticks of dynamite or whatever. And rIIA, the sensor, the trigger, conveniently looking like a little radar dish, the detector that detects whether somebody's infected me. And one of the beauties of stories in molecular biology is that science actually works. The details of papers that were published 30 or 40 years ago still matter and can still be used. And in this case, my mentor, David Parma, had worked out very laboriously that rIIB was probably composed of 4 transmembrane helices. This is a figure from his paper. And by clever experiments that were possible at the time, he knew which bits were inside the cell and which bits were outside the cell. He knew the topology. So now AlphaFold in seconds basically gives me that 4 transmembrane helix structure, and because of but does not tell me which direction that goes in the membrane, and it's because of David's experiments from 1992 that I know which direction that goes in the membrane. What David would have really liked to have lived to see is what rIIB, that is not a proton channel. That thing is actually just a tight 4 helix bundle, so by itself can't be a channel. But what I can do is I can use AlphaFold to say, try to make it a trimer, try to make it a quadrimer, try to make it a pentamer. What do you think this thing is? The moat A engine of the bacterial flagella is a pentamer, it's a 5-mer. And so by homology, by sequence analysis, my guess is that rIIB makes a bundle of 5, and that's what AlphaFold thinks too, and it's a beautiful structure that I wish I could show to David Parma. So this is it on its side, that's a single subunit of rIIB. That down the middle is what I'm guessing to be the proton pore. Mind you, this is a prediction. This would need to be confirmed by experiment. We don't completely trust these programs, but a beautiful symmetric structure. That is probably the bomb. That hole through the middle is, when activated, what puts protons through and kills E. coli. Now, complicated slide to make a point that Rich also made. Another thing that has happened, actually just in the last few years, is that people have realized that rIIB, this defense system from Lambda that Crick and Brenner were based on, is the granddaddy of a whole bunch of defense systems. So there are now on the order of 200 to 300 known different molecular weaponry systems that bacteria are using to defend themselves against viruses. Some of them are famous. You know about CRISPR. CRISPR is one of these defense systems. CRISPR has now been harnessed for genome editing at scale to treat patients by directly changing bases in their genome that need to be changed. You may also know about restriction enzymes that are also, so this was the fundament of molecular biology in the 1980s, being able to cut and paste DNA together to make recombinant DNA. These things are examples of us discovering weapon systems in this host virus warfare and then making use of them as molecular tools. And the excitement of finding that those are just 2 and Rex is another, and that there's 200 or 300 more, part of that excitement is there are probably other really cool tools that we can get from taking advantage of this highly honed evolution. So the way that people are discovering those, and so I just want to close with one more story and one more complicated slide that I'll walk you through, is that they're using sequence analysis of pretty much exactly the type I talked about. It happens when you look at across the top that when weapon systems are deployed by the host against viruses, they bundle them up into what's called defense islands. They will have multiple defense systems at the same time. And what that means is if you know already, because of work in the 1980s about restriction modification systems, that's a type 4 one and that's a type 1 one, you can use what the field calls guilt by association. You can say, there's other proteins in this defense island that I don't know what they are, but I can tell from the PFAM database and from the domains of unknown function that there is a protein family there that has been observed to be conserved across lots and lots of bacteria. And it's recurring lots of times with other known weapon systems. It must be a weapon system. Let's clone it out and see if it defends against phage. And that's been an extraordinarily fruitful way of discussing. That's why we now have 200 or 300 of these. And this is one of them. And the reason it caught my eye in one of these papers was probably because I read these papers because I love to see people take advantage of the domains of unknown function. It's been very gratifying. But there is the MOTE-A channel. This is the first defense. So the REX, what I told you about REX has not been recognized in the community. But this is the first one where somebody saw that there's a weapon system that has homology to MOTE-A. So this, to me, means that there's an additional clue. But what I want to leave you with here is a story that this is yet another deployment of the MOTE-A homology, in this case, in a very weird and complicated situation. That's the transmembrane region. If you tilt it up on its side, ZOR-A is the homolog, and there's 5 of them as you go around. So that's the homolog to what I'm thinking about. But there's a top that's ZOR-B, and then there's a tail on this thing that is 70 nanometers long. That's actually a substantial part of the size of E. coli. E. coli is only about 500 nanometers across. So whatever this spiky thing is a big spiky thing. And their crazy model is that this is indeed still a proton motor, and that when the phage injects, the ZOR defense system starts spinning and wraps the DNA, I don't even think this is physically possible, but it's lovely to look at, wraps the DNA around and then attacks the DNA and chews it up like a little cotton candy getting eaten. And there was a paper I just saw this morning that's just come out on the bioRxiv as a preprint. This is a super hot area where I think the connection to the rIIB proton channel might end up being informative, and that's the kind of game I play. Now, I'm sorry to tell you, I have to give a disclaimer. Everything that I just told you has been declared by the National Institutes of Health to be of utterly no value to the US taxpayer, and all of my funding was terminated last May on that basis, partially restored, they're still suing us, blah, blah, blah. So I'm very sorry that I've been wasting your money on this stuff. The picture on the right is a conversation between me and one of the engineers at Google DeepMind, one of the AlphaFold 3 team, asking us for updates on the software because we're closely tied, and we can't currently provide them that because our project is temporarily on hold while I guess we wait for midterm elections. But I don't want to leave you on that note. The note I want to leave you on is that this is an amazingly fun and productive game that we get to play with genome sequences. Comparative genome sequence analysis, though it doesn't get you everything you want from biochemistry and structure, it guides the experiments. It gives us important clues. It lets us move between model systems and human to move information from genome to genome. It's a lovely thing to be a part of, and regardless of the state of US funding of science, I'm going to keep doing it.
Well, thank you, Sean. No, it didn't. Thank you for that, and thank you for your remarks at the end, because it just shows you what we're living in right now, and I'm very gratified to hear that you're going to continue anyway, right? Fuck 'em, okay?
Exactly.
Good. Pleased to hear it. My feeling exactly, but I'm going to use the moderator's privilege here and ask you a question about a future question. So, we hear a lot about AI, and some people who are very well respected life science scientists think it's all a nonsense and it's not going to make all that much difference to what we do. I want to ask you the question, having been through the recombinant DNA revolution and a lot of technologies since then, do you think that we're now at a stage that is not dissimilar to where we were in 74 and 75 with the advent of restriction enzymes and what you can now do with them when you look at AI and the power of what it can do? You got your mic.
Yeah, I got it. We're absolutely, so the problem with these things is when a new technology like this comes in, you can get a lot of airtime by poo-pooing it. It's like, look at all the stupid errors that ChatGPT makes, ha ha ha, which it does, which I love to tell, this is what we sit around at lunch and here's this latest stupid thing that ChatGPT did. And you can also like oversell it. You can say like everybody's going to be out of work, we're going to be replaced by AI robots, and you can give a lot of talks on this. Usually when these kind of technologies come in, recombinant DNA, DNA sequencing, whatever. It actually is a revolution, and you've got to keep working on it, and you've got to keep patching the things that aren't working and keep working on the things that are working. And so the answer to your question is yes, we are absolutely in a revolution right now. AlphaFold is a particular example where I can sit on my laptop on a plane and feed a sequence into AlphaFold and get a structure that's probably right in 5 minutes. And that is just an unbelievable technological advance.
Yeah, I think it's amazing, and I have changed my mind. I never thought I would say that, but I absolutely agree with you.
That was just beautiful. I'm so glad that you put into proper context the difficulty of really getting multiple sequence alignment right. And so for those of us who have looked at related sequences, it's not a trivial problem, especially when you go from something that has 4 letters like nucleic acids to 20. So going from alignment to then identifying probability of proximity of 2 amino acids is what led to AlphaFold success. So now coming back to the nucleic acid end, what are your thoughts about taking advantage of this much more obvious probability of proximity in DNA and RNA of base pairing to determine structures of nucleic acids?
Thank you for this question, Naboisha. This is a setup. I will give you $1 later, buy you a beer. What I actually work on most of the time is RNA structure. And so I showed you a software package called Hammer that's for linear sequence analysis that I use as the warm-up project for what I really care about, which is how RNAs fold into 2D and 3D structures. And the base pair correlations in RNAs, as you just said, are super strong. One very interesting thing is that whatever AlphaFold is doing, so AF3 will now take RNA as input, and in contrast to what it does with protein, it is, in my opinion, laughably bad with RNA. It comes back with predictions for RNA that it says it's completely confident of, and it's just like, that isn't even physically reasonable. It's like they are still far off on RNA, which is interesting because those base pair correlations are way, way stronger than what we see in protein. It's really obvious where the base pairs and even the triples and the tertiaries and what have you are in RNA. AlphaFold, for whatever reason, is not picking that up, whatever, and so there's a gap there, and people are working on that. So my partner, Elena Rivas, is working on it directly with an open source clone of the AlphaFold code called OpenFold, where they're trying to make the RNA version and learn from that. And I'm coming at it from the standpoint of, can we use other machine learning methods and specialize them better for RNA structure, working sort of outside the AlphaFold framework. But so the short answer is that remains, interestingly, an open problem, even though Gary Stormo and others introduced covariation analysis back in the day. The AI methods are not using it effectively as far as I can tell.
One over there.
Wow, so this is a very interesting comment on something that was even not part of your talk. So I would love to follow up offline. I have a back to proteins question. So coming from the sequence side, you're arriving at 30,000 or so PFAM domains. Coming from the structure side, there are about 1,000 folds. So could you comment on the importance of structural variability, I would say, within a fold that would then reflect and map back to your much larger number of domains in PFAM?
Exactly. So this is sort of a double-edged question. On the one hand, that is exactly right. The 30,000 protein domains, many of them we can recognize once we know structure, or these days, having predicted structure with AlphaFold. We can recognize that they have structures that are so similar that they must be homologous, but the sequences have just diverged so fast that we can't recognize it by sequence. People like me working from the sequence end are trying to make that better and better. We're trying to push deeper and deeper in evolutionary time to recognize those kind of similarities, but having the structure lets you see it. And so PFAM calls these clans. We'll group families into clans where we say we know from structure that these are probably homologous, We just can't see it with a sequence. And that's an important organizational principle for understanding how to move information from domain to domain. You know, you've got some RNase domain that you have some structurally related RNase domain or something. You go, evolutionarily, those are probably related to each other. But then the other side of this edge is the structure comparison is so good that especially for simple structures, you worry about convergent rather than divergent evolution. So that little REXB 4-helix bundle, I can get it to align to other 4-helix bundles, but 4-helix bundles are probably dime a dozen. So does that really tell me that it's divergently related by common ancestry to those things, or is that just a structural motif that happens over and over again? We don't have that mathematically grounded. People will use subjective criteria to try to distinguish convergent from divergent evolution. Sometimes it's not even clear that it matters. If something convergently evolves to the same structure because it's being forced into the same function, then that's still telling you what we need to know, which is something about the shared function. So it gets a little murky as you get this far out that you're relying on structural comparison, whether you're still looking at evolutionary ancestry or just something that's been forced into the same piece of structure space.
Okay, I'm going to add to that in the sense of asking you a question about David Baker's work, which was beautifully summarized in a paper in Nature a couple of weeks ago. You say 30,000 folds, but actually there are many more thousand folds that you can make when you use some of the new techniques. Can you comment on that?
Oh, I mean, that's an interesting question too. I mean, you take the number, there's 20 amino acids, and if I just say 200 amino acid sequences, so I just make it a fixed length, there's 20 of the 200 sequences. That's an enormous number. And then you imagine that sort of laid out as a space or, you know, a 2D map of the protein universe. These protein families are like little galaxies, little clusters in that space. And so evolution has thrown its darts of de novo gene origination into that space. And once you hit something that works, you tend to stay there. Right. And now with synthetic biology, we can throw new darts into that space and maybe access parts of the space that evolution just hasn't even had time. I mean, it's been a lot of time, but probably not an infinite amount of time to completely sample a space that big. So there probably are other sort of places you could put a galaxy where evolution just hasn't visited yet.
And I don't think we should underestimate the importance of that in making new proteins to do new things, which we need to do as a species. At the back.
So, the idea of the convergent versus divergent evolution of three-dimensional structures, that's a very age-old question. I think it was about in the 1970s somewhere, Walter Fitch wrote a paper basically laying out at least the intellectual framework for how to answer that, which was to, you know, align the sequences of the different, I guess, clan members, I guess would be in your terminology, and do ancestral reconstruction back to internal nodes. And if there was more similarity at the internal node versus the external nodes, then that would be an argument for divergent evolution rather than convergent evolution. Have you tried that?
We don't, but I don't, but there are people who do that. And so that idea and ideas like it have been picked up. One of the attempts to sort of formalize divergence versus convergence that's even more recent than Fitch is a guy named Doug Theobald, who I think is still at Brandeis, and has got that idea, and also the idea that what you expect from divergence is congruence with the species tree. So now that we're pretty, you know, sort of set on what the species tree is, you expect divergent evolution to follow that same tree gene after gene after gene for the most part. There's a lot of horizontal transfer and stuff like that. Now, neither of those ideas really works at the distances that I'm talking about. We're down to sequences. Hammer can detect significant relationships between sequences that are like 5 to 10% identical to each other. And at that point, the ancestral sequence reconstructions are really uncertain.
One there.
The AlphaFold 2 was trained on a kind of oil, I think about it, which was all these crystallography, billions of dollars spent creating these structures, cryo-M. And it seemed like the next frontier that you mentioned is, okay, now we have these structures, you know, they're mostly correct. What do they do? What is the oil for that problem? Like, what is the data that's missing so that the next version of the AlphaFold that's actually working on the function, not just the form, is able to be trained?
I don't have a good answer for that. I know from just talking to other people in the sort of high throughput pharma kind of community that there's a lot of efforts to do high throughput perturb-seq and stuff like that to sort of generate phenotypes at scale and hook them up to deep learning. But in my mind, those are often very sort of far from the sort of biochemical and structural detail that I want as a biologist. And so I'm still a proponent of we still need to do the work. We still need to go in and understand things like loving directed mutation at a time of like this residue is probably the catalytic residue doing this, and that requires just a ton of integration of knowledge that I don't know how to generate that kind of knowledge at scale to use as oil to feed into the pipeline for something like a future AlphaFold. I think we still need biology.
I think that's a great place to leave this session, actually. Not that we have run out of time, but actually that's a good way to stop